Medical experts agree that our genomes will play an important role in our healthcare futures in the prevention, diagnosis, and treatment of disease. To make this future a reality, one of the challenges we have is how to create structure from unstructured health and genomic information, and then to integrate this with an individual's own genomic profile so we can have a meaningful impact on their healthcare choices. At Station X, we believe in this future and are already creating the software to power it. We call it GenePool. In this short video, we'll show you how GenePool is already helping people make discoveries from very large amounts of genomic data. Before we show you that, a little background might be useful. Firstly, what is a genome? It's the complete set of genetic instructions encoded in the DNA that you inherited from your parents. Each cell has a copy, and it's stored as long strings of the bases A, G, C, and T. Altogether, there are 3 billion of these bases. Every cell has a copy of your genome, and the instructions it contains guides each cell as to which proteins it should make. At the genomic level, each person is 99.9% similar to every other person. That may sound close, but there's still 3 to 4 million differences between you and anyone else, and these differences are what make you unique. Most of these differences don't have any obvious impact on you. Some of us may have variations that are responsible for well-known traits and disorders such as cystic fibrosis. But still other variations might be related to much more rare disorders. By sequencing the genomes of people with these rare disorders, scientists can identify the genetic basis of that condition. Over time, some of our cells may accumulate additional genetic changes for various reasons, for example through chemical damage to our DNA. These mutations can adversely affect the normal production of proteins, turning these cells cancerous. Scientists are using genomic sequencing to compare the genomes of cancer cells to normal cells. In this way, they can identify patterns of mutation and gene expression that characterize the cancer. If these patterns are reproducible, they can be used to develop diagnostic tests and therapies that are specifically targeted to that cancer. And that's where GenePool comes in. It's a platform for clinical researchers to easily manage, analyze, and share their large-scale genomic data, and it greatly shortens the time it takes for them to get meaningful results.
To analyze the genome of an individual in GenePool, we will first retrieve the sequence variants or changes in their DNA in the protein coding or exome regions of the genome. We'll then look at some of the visual features of GenePool to filter those down to interesting variants. First, we will look for changes that have known clinical significance and select pathogenic. We're looking at 26 variants here in a number of genes, but we also want to remove variants that are common in European populations for this individual because those are unlikely to be disease-causing. Let's take a look at this first one in the sequence itself. So this view in the gene browser of GenePool shows the evidence for that variant. And in this case, a little over half of the reads have that variant and show a change in the amino acid sequence. Now let's return and use a slightly less restrictive method for identifying potentially interesting variants in this genome. First, we're going to relax the filter for the clinical significance and apply a different filter for variants that affect proteins. We'll then go back to clinical significance and select 3 different categories that might be potentially clinically interesting. We're now looking at 28 different variants, which we can summarize by gene, and in fact go in and look for genes that might be affected by 2 different variants, each inherited from a different parent, called compound hets. Let's go look at this gene, which is an example of one of those, and zoom in on the region where each of those variants occur, one here on the left-hand side and one here on the right-hand side. In this GenePool example, we're going to look at a prostate cancer study in which 20 individuals with prostate cancer are compared to 20 individuals without prostate cancer to identify genes that behave differently in these 2 groups. In GenePool, you can simply drag and drop your samples into each category and run the analysis to identify genes that behave differently in these 2 groups. Each of the visual widgets are also used as filters, in this case to find the most significantly differentially expressed or used genes in these samples. Each of the visual and tabular views are interactive. By selecting any gene on the table, you can drill down and see how each gene is used in any of the cancer or normal samples. You can also view the expression or use of that gene across its structure on the genome. In this case, comparing the read depth or the sequencing depth and patterns across those cancer and normal tissues. In addition to looking at summary level views, we can zoom all the way in to look at the sequence reads that support the measurements for these genes. Now, returning to the original table of our most differentially expressed genes, we can ask what biological pathways or functions are common in these pathways. We can also ask how these genes are related to each other and produce a network of relationships and zoom in on individual genes to see how they are connected to other genes in our list. Lastly, a very important feature of GenePool is the ability to save your results or export them so that you can share them with your colleagues at a later time.
Lastly, thank you very much for your time. We hope you've enjoyed this brief overview of GenePool. If you'd like to learn more about it, including getting a trial account, or if you'd like to hear about investment opportunities at Station X, please drop us a note to these emails. We look forward to hearing from you. Thank you.